In this video, we're going to start discussing the third key area from the human cells, part of the higher human biology, which is all about gene expression, building proteins from the information contained in DNA. So we'll start here by looking at the different types of RNA and their structures. So first off, what do we mean by gene expression? Okay, so expression is the active building of a protein based upon the genetic code, the sequence of bases that appear within the DNA. This is going to involve two big processes: transcription and translation. Okay. Remember, each cell has the entire DNA molecule within its nucleus, but it's actually going to express or build a fraction of those proteins. Our specialised or differentiated cells are only going to build the proteins that are relevant to their function, so only a small number of genes will actually be expressed. Cell has another big problem. Its DNA must remain in the nucleus at all times. It has to be protected, kept safe. But the site of gene expression where the proteins are built is the ribosome. Okay, those ribosomes are within the cytoplasm. So DNA can't leave the nucleus, but proteins are built in ribosomes. How do we get that information where it needs to be? So that's where the involvement of this intermediate molecule, the RNA, gets involved. And so there's three different types: the messenger or mRNA, the transfer or the tRNA, and finally the ribosomal, the rRNA. So in terms of RNA structure, all three types are nucleic acids, which means they will have some features in common with the DNA molecule that we already discussed in the DNA structure video. So because they're nucleic acids, it means they're going to be made up of those nucleotides, the base, the sugar, and the phosphate group. Major differences this time is that the three types of RNA are all single-stranded, so a single strand of repeating nucleotides versus the double-stranded nature of DNA. Another difference is the type of sugar, so this time it's a ribose sugar. And the four bases, we still have C, G, and A, but in all the types of RNA, the T is replaced by uracil U. But other than that, base pairing rules still apply. RNA, DNA, very similar molecules, but with these differences. So if we start by looking a little closer at the messenger or mRNA, so this is the first type of RNA that you will come across in the process of building a protein or doing gene expression. So the message that the messenger RNA contains is the information on how to build that protein that was found within the DNA molecule. So transcription is the process of building the messenger, the mRNA, and this takes place within the nucleus. The DNA is safe, it's protected, so we build the mRNA in the nucleus using the information found in the DNA molecule. So apply your base pairing rule. If there's a C on the DNA, there'll be a G in the mRNA and vice versa. If there is a T in your DNA, you can still add an A onto the mRNA. A is still present. But the difference in bases, if there's an A on the DNA, there's no T in the RNA to add, so U will be added in place. So, faithful copy of the DNA molecule using the base pairing rules. Another key feature that will be helpful when it comes to looking at translation is to remember when you read the mRNA molecule, you'll read it three bases at a time. So every 3 bases is known as a codon, and what it codes for is one specific amino acid. You'll always be able to work out which amino acid using one of these codon charts. The T, or the transfer RNA, this will be important when it comes to the translation process of gene expression. Again, it's another one of these intermediate RNA molecules. It is still single-stranded, whereas the mRNA was this sort of string or straight line of nucleotides. The tRNA actually begins to fold and bend and loop, but it is still one continuous line all the way through. Okay, so still a single-stranded molecule. Two features that are very important is firstly the anti-codon site, so 3 bases right down at the bottom here of the tRNA molecule. So the anti or opposite to the codons of the messenger RNA. So base pairing rule is going to become important again. If there's a G on the codon, there must be a C on the anticodon. There's an A here, so a U in the anticodon. G here, so a C on the anticodon. It's complementary and that allows the attachment or binding of the tRNA to the mRNA. The other important site is at the opposite end, your amino acid attachment site. So at one end it will bind to the messenger RNA, and at the opposite, bring in the corresponding amino acid into the ribosome. And finally, just a quick word about the rRNA. It's a structural component, so it actually makes up the physical structure of the ribosome where protein synthesis is taking place. So you can see it here in the diagram. The messenger RNA comes into the ribosome. It's read 3 bases or a codon at a time. And you can also see the tRNAs, they will also enter into the ribosome, bind to the mRNA at their anticodon, and bring in the corresponding amino acid at the opposite end, which eventually is going to build up the protein. If you want more details on how we build the mRNA and then build the protein, there will be subsequent videos on both transcription and translation. And for now, let's try a past paper question all about the structure. So you're asked to identify the RNA codon and the anticodon if the DNA base sequence is GAT. Pause the video, select your answer, and restart to find out if you were correct. So let's work it out together. If there's a G on the DNA, that means a C would be added to the mRNA. If there's an A, a U, and T, and A. So that must be your codon with 3 bases added to the mRNA. So must be C out of these options. And we can confirm by thinking about, okay, if that's a codon, then what must be on the tRNA molecule that would bind? So if there's a C on the codon, that means you would find a G in the anticodon, a U would be an A a U. Okay, so it has to be C. It's the only one that works out. Okay. So next videos up be transcription and translation to let you see a little bit more about how these molecules that we've discussed in this video actually go about building the protein. Any questions, any queries you can leave in the comments below.